red blood cell and platelet preservation, and the historical perspectives and current trends. So some historical background. Back in 1492, it was the first recorded attempt at a blood transfusion. This involved Pope Innocent the Seventh. The procedure was the blood from 3 young men was transfused in an attempt to try to revive him. The outcome was not great. All 4 people actually did die. The significance of this is that it marks the beginning of the concept. Even though it was unsuccessful, it still kind of brought up, can it work? So they had some major obstacles to overcome in transfusion therapy. This is to make blood transfusion safe and effective. So here are some things that we have done. So we need a non-toxic anticoagulant. The purpose of this is to prevent clotting of blood outside of the body. Some early issues, the use of substances that were, they were toxic or they were ineffective at what they were supposed to be doing. So the solution was a development of a safe anticoagulant like sodium citrate back in 1914, which actually binds the calcium ions and that prevents the coagulation of the blood. Another thing was to have the appropriate devices to perform the transfusion. Early attempts were often fatal, as mentioned earlier, and kind of crude. So advances as we now have sterile equipment, accurate control of blood flow rate, and we're also able to cross-match for compatibility. We also needed appropriate preservation solutions. This was needed to extend the shelf life of the stored blood. So we had the development of preservations like citrate phosphate dextrose, or CPD. The addition solutions to maintain red cell viability, so it's able to live a little bit longer on the shelf than previously. We need to also avoid circulatory overload, which is a condition where the circulatory system is overwhelmed by just too much fluid being put in the body. So the prevention is having a careful volume control during the transfusion and monitoring patient vitals and fluid status. We also need a component therapy, which is a transition from whole blood transfusions to component therapy like red blood cells, platelets, plasma, and cryoprecipitate. The benefits are it was a targeted treatment. We also are able to be more efficient at the use of donated blood instead of giving the whole thing to everyone. Not everyone needs the whole blood. It also helped to reduce risk of complications. The donation process, so we have education materials. The purpose is to inform potential donors about the process itself, any risk and benefits that could happen, criteria for eligibility, and behaviors or conditions that may defer donation. The content includes transfusion, transmissible infections, so HIV, hepatitis, that type of things. Also includes travel-related risk, so like malaria areas, any medication that a patient may be on, and post-donation care. Requirements, so it must be reviewed before donation, and this helps to ensure that there is informed consent and safety. The donor also fills out a health history questionnaire. The comprehensive survey will assess medical history, travel history, high-risk behaviors, vaccination status, as well as any past surgeries or transfusions. The goal of this is to help identify potential risks for transfusion transmissible diseases and to ensure the safety of both donor and the recipient. This must be completed at each donation every time they donate. So abbreviated physical examination will also be performed. This is performed on site before the donation, and this usually will include the temperature check, pulse rate, blood pressure, their hemoglobin or hematocrit level, and the patient's weight. This helps to ensure that the donor is medically stable and able to donate. They also make sure that they are not anemic and that they are within the safe parameters to donate blood. These are the current donor screening tests for infectious diseases. So red blood cell biology and preservation. So the normal red blood cell lifespan is about 120 days in the body, so in the circulation. Its function is to transport oxygen and carbon dioxide efficiently throughout the body. The survival is going to depend on the integrity of key, these areas. So the red blood cell membrane, hemoglobin structure and function, and red blood cell metabolism. The red blood cell membrane is essential for maintaining the cell shape and deformability. It allows the cell to survive the physical stresses of, going through circulation. And it also helps to regulate the exchange of ions and gases. It functions as a semi-permeable lipid bilayer, enabling selective transport and maintaining osmotic balance. The structural composition, it has a lipid bilayer. The main component of that is a phospholipid. It is asymmetrically distributed across the membrane, and it provides fluidity and barrier function. Also, it contains cholesterol, which helps modulate membrane stiffness and fluidity. The cytoskeleton is a protein mesh-like structure that supports the lipid bilayer, and it also includes certain proteins like spectrin, ankyrin, actin, and protein 4.1 and 4.2. The role is to provide a mechanical strength, maintains biconcave shape, that donut shape of the red blood cell, and also allows deformation as red blood cells pass through the capillaries. The membrane proteins are integral proteins. They span the lipid bilayer. Some examples of those are glycophorins, which help carry blood group antigens. The function is to transport molecules. It helps to provide anchoring points for the cytoskeleton as well. These peripheral proteins attach to the inner or outer surface of the membrane, and the function is a structural support, signal transduction, and interaction with the cytoskeleton. Some clinical relevance is there, if there's any defects in the membrane proteins or lipid composition, it can lead to certain conditions like hereditary spherocytosis, elliptocytosis, and acanthocytosis. These disorders can cause increased red blood cell fragility and shorten lifespan for the red blood cell, which could lead to hemolytic anemia. The asymmetrical organization of the red blood cell membrane. So the lipid bilayer of the red blood cell membrane is asymmetrically arranged. This means that the specific lipids are localized peripherally to either the outer or inner leaflet of the membrane. So the outer or external layer is composed mainly of those glycolipids and choline-containing phospholipids such as phosphatidylcholine or sphingomyelin. The functions of these are for surface antigen expression, so your blood group antigens, which we'll get into later on in this course, and also interaction with the immune system and membrane stability and cell recognition. The internal or cytoplasmic layer is composed mainly of aminophospholipid. The functions of these is a membrane curvature, signal transduction, and phosphatidylserine exposure is a marker of cell aging or apoptosis and flags the cell for removal from the body. Biochemical composition of the red blood cell membrane. So the red blood cell membrane is composed of three major biochemical classes. 52% of that is protein. So we have the integral proteins and the peripheral proteins, and these help to, as mentioned earlier, provide structure, transport, and antigenic properties. 40% is lipids, so phospholipids, cholesterol, and glycolipids. Like I mentioned earlier, these maintain the fluidity, permeability, and asymmetry. 8% are carbohydrates, and these mainly are attached to proteins and lipids as glycoproteins and glycolipids, and these are important for cell recognition and antigenicity. So some clinical relevance, any disruption of a lipid asymmetry, like exposure of any of the lipids on the outer leaflet, that's a sign of red blood cell aging, apoptosis, which is self-destruction essentially of the cell, and hemolytic anemias. An imbalance in membrane composition can contribute to red blood cell deformability issues, immune clearance, and membrane fragility. Red blood cell deformability. So the importance of deformability in red blood cells is so basically deformability is the ability of red blood cells to change shape without rupturing as they pass through the narrow capillaries and the splenic microvasculature in the body. This property is critical and crucial for efficient oxygen delivery, survival in circulation, and helping to avoid premature destruction. So role of ATP in the membrane flexibility. ATP is essential for maintaining membrane integrity and flexibility through a couple of different things. So one is phosphorylation of spectrin, which is a key cytoskeleton protein, and active ion transport, so those sodium, potassium pumps, and those calcium pumps as well. Loss of ATP will result in reduced spectrin phosphorylation and loss of cytoskeletal flexibility. It can also decrease that membrane deformability. Role of calcium in the membrane rigidity is in healthy red blood cells, intracellular calcium levels are tightly regulated. Increased membrane calcium can lead to activation of calcium-dependent proteases, can also do cross-linking of cytoskeletal proteins and membrane stiffening, so it makes it kind of more, rigid, not able to bend and flex as well. Clinical consequences of splenic sequestration, my goodness, I'm sorry. So it can be because of rigid or poorly deformed red blood cells. These are going to be trapped in the spleen. They're going to be recognized as abnormal by the splenic macrophages, and then they're going to be phagocytized and removed from circulation. So the result of that is we can have extravascular hemolysis and potentially splenomegaly. Deformability, once again, so the loss of red blood cell membrane as seen in spherocytes and bite cells will shorten the survival of these forms, and I just have a picture on the side there that kind of shows both of these. So permeability, the red blood cell membrane, as mentioned earlier, plays a crucial role in maintaining the cell volume, osmotic balance, and prevention of hemolysis. And this is achieved through its selective permeability and active ion transport mechanisms. Passive permeability of the red blood cell membrane basically is for a freely permeable red blood cell to go back and forth with water and ions such as chloride and bicarbonate. And these allow osmotic equilibrium and also helps in the participation in gas exchange and acid-base balance. You can have impermeable to cations, so the sodium and potassium. So passive movement of these ions is highly restricted to preserve intracellular electrolyte concentration. The active transport mechanism, so this is to maintain intracellular ion gradients. The red blood cell membrane contains ATP-dependent pumps, which include the sodium-potassium ATPase. This maintains the high intracellular potassium and low intracellular sodium levels. This prevents osmotic swelling and lysis by controlling that cation balance within the cell. Your calcium ATPase actively pumps calcium out of the red blood cell, and this keeps your intracellular calcium levels extremely low. It helps to prevent membrane rigidity and premature cell death. So clinical relevance of all of this is a failure of active transport can lead to cation imbalance, osmotic swelling, and hemolysis. And this can be seen in conditions like enzyme deficiencies, oxidative damage, and aging red blood cells. Red blood cell metabolism, so they have a unique metabolic profile of red blood cells. The red blood cells have a highly specialized and limited metabolism due to their structure. They are anucleate, which means they have no nucleus, and they lack a mitochondria. So as a result, red blood cells cannot perform oxidative phosphorylation and rely exclusively on the anaerobic pathway for energy production. ATP production in red blood cells, they follow the anaerobic glycolysis or the Embden-Meyerhof pathway. Primary source of ATP in red blood cells this is how they get it. It converts glucose to lactate, which then produces 2 ATP molecules per glucose formed, and NADH, which contributes to that redox balance. It supports membrane ion pumps and maintaining deformability in volume and preservation of the redox environment. Secondary pathways, not for ATP, but for vital survival, they use the hexose monophosphate shunt, which produces NADPH to protect against the oxidative damage, and methemoglobin reductase pathway, which maintains hemoglobin in its ferrous oxygen-carrying state. Also uses the Lou-Behring Rappaport shunt, which produces the 2,3-biphosphoglycerate 2,3-BPG, which modulates hemoglobin oxygen affinity. So why don't they have oxidative metabolism? They don't have a mitochondria. That prevents oxidative phosphorylation. It limits oxygen consumption, which ensures oxygen is delivered to tissues not used by the red blood cells. Because of their anucleus nature, red blood cells cannot synthesize new proteins or enzymes, so their lifespan is about, like we mentioned earlier, about 120 days. Clinical relevance of this, so if there's any deficiencies in the metabolic enzymes like that pyruvate kinase, it can lead to impaired ATP or NADPH production, which can then lead to hemolytic anemia. ATP depletion also leads to loss of deformability, ion imbalance, membrane damage, and premature red blood cell destruction. Hemoglobin dissociation curve. So the primary role of hemoglobin is it is the oxygen carrying protein found in the red blood cells, and it performs two essential physiological functions. It delivers oxygen to peripheral tissues, and it will transport carbon dioxide from tissues back to the lungs for exhalation. So hemoglobin and oxygen binding, each hemoglobin molecule can bind up to 4 oxygen molecules. The binding of oxygen is cooperative. So binding of one oxygen molecule increases the affinity for the next. This results in a sigmoid shape or an S-shaped oxygen dissociation curve. The oxygen dissociation curve, like mentioned earlier, is a sigmoid or S-shaped curve, and it reflects the cooperative binding of oxygen, and it allows hemoglobin to efficiently load oxygen in the lungs, which is a high partial oxygen, and release oxygen in tissues, which is a low partial oxygen. The purpose of blood preservation. So the primary goal of blood preservation is to ensure the availability of viable and functional blood components for patients who need blood transfusions. Red blood cell viability refers to their ability to survive in circulation and perform their intended functions, which is oxygen delivery and carbon dioxide removal. It is crucial that red blood cells retain their normal shape, membrane integrity, metabolic activity, and hemoglobin function during the storage. So some considerations that we need, during storage is that red blood cells are typically stored at 1 to 6 degrees Celsius in an anticoagulant preservative solution. Storage time is usually up to 42 days, and it must make sure that we preserve the membrane deformability, ATP levels, the 2,3 BPG levels, which affect the oxygen release, and low levels of hemolysis. We don't want that to happen, or we don't want hemolysis to happen, so we want low levels. The clinical importance is we have transfusing non-viable or damaged red blood cells can actually impair oxygen delivery and can cause immune reactions or complications to the person receiving the blood. So we need to make sure that we are monitoring the storage conditions and shelf life for patient safety. The FDA, or the Food and Drug Administration, has certain requirements as well for red blood cells. So the average 24-hour post-transfusion red blood cell survival must be greater than 75%. So this means that at least 75% of transfused red blood cells should still be circulating 24 hours after transfusion. Also, free hemoglobin in storage must be less than 1% of total hemoglobin. So the low free hemoglobin indicates a minimal hemolysis during storage. Red blood cell viability. So the loss of red blood cell viability has been correlated with the lesion of storage, which is associated with various biochemical changes. It's listed there on that chart, so you can read if it decreases or increases the red blood cell viability. Red blood cell storage and the 2,3 DPG levels. So as red blood cells are stored, the 2,3 DPG levels decrease. The DPG depleted red blood cells may have an impaired capacity to deliver oxygen to the tissues. The 2,3 DPG is reformed in stored red blood cells after in vivo circulation, and this will depend on the recipient's acid-base status, their phosphorus metabolism, the degree of anemia the patient has, and the overall severity of the disorder. Anticoagulant preservation solutions. So incorporation of adenine increases the glycolysis and ATP levels. The pathophysiologic effects of the transfusion of red blood cells with low 2,3 DPG levels and increased affinity for oxygen can increase cardiac output, can decrease the mixed venous tension, and it can also just cause a combination of these. Additive solutions, they are preserving solutions that are added to the red blood cells after removal of the plasma with or without platelets still in there. It can help to extend the shelf life of red blood cells to 42 days by adding these nutrients. It can also allow for the harvesting of more plasma and platelets from the unit, and it can also produce a packed red blood cell of lower viscosity that is easier to infuse into the patient. Currently, 3 additive solutions are licensed in the United States for use. The first one is Adsol, next one is Nutricel, and the last one is Optisol. All of these additive solutions are approved for 42 days of storage for packed red blood cells. None of the additive solutions maintain the 2,3-DPG throughout the entire storage time. So as with red blood cells stored only with primary anticoagulant preservatives, my goodness, I'm sorry, the 2,3-DPG is depleted by the second week of storage. Red blood cell freezing, so primarily is this used for patients that have autologous units and the storage of rare blood types. You can also have a cryoprotective agent that is going to be added to the red blood cells that are less than 6 days old. Glycerol is used most often and is added to the red blood cells slowly with vigorous shaking, and that helps to enable the glycerol to permeate the red blood cells. Red blood cells are then rapidly frozen, and they are going to be stored at negative 65 degrees Celsius. Again, this doesn't happen too often, but it can. So currently the FDA licenses frozen red blood cells for a period of 10 years from the date of freezing. Transfusion of frozen cells must be preceded by a deglycerolization process. So we have to remove that glycerol, and that is achieved by systematically replacing the cryoprotectant with decreasing concentrations of saline. Excessive hemolysis is monitored with the hemoglobin concentration of the washed supernatant. Osmolality of the unit should also be monitored to ensure adequate deglycerization before given to the patient. So platelet preservation. So annually in the U.S., millions of platelet units are distributed and transfused. The financial impact of outdated and returned platelet units is a primary reason that we need to improve inventory management. There is significant wastage of platelet products. Platelet preservation is a key strategy to reduce the number of outdated and discarded platelet units. So there are a lot of these that they get collected and they actually don't get used. So that's where that waste part comes in. Platelet storage, as mentioned earlier, is a major challenge to the blood bank because it has a lot of storage limitations. It only has a 5-day shelf life in the United States. Bacterial contamination can occur because platelets are stored at room temp. There are varying degrees of platelet activation and aggregation, release of intracellular granules, and there can be a decline in ATP and ATP levels for platelets as well. So also for platelet storage, they can have the loss of platelet quality as it's sitting in storage as well. Quality control testing of platelet units is essential for ensuring that only viable and functional platelets are transfused. These tests help to reduce wastage by identifying units that no longer meet standards and improve patient outcomes by guaranteeing the efficacy of transfused products. For clinical use of platelets, we use these for the treatment of bleeding associated with thrombocytopenia. So somebody who has a decreased platelet count may need some platelets. Can also be used as a prophylactic treatment for hematology oncology patients with thrombocytopenia as well. Today, platelets are prepared as concentrates from whole blood and increasingly by apheresis. Platelet concentrates are prepared from whole blood, and apheresis components are routinely stored at that room temp, 20 to 24 degrees Celsius, with continuous agitation or movement for up to 5 days. FDA standards define that expiration time as midnight of day 5. That way it's consistent for everyone. In the United States, platelets are being stored in 100% plasma medium unless there is a specific platelet additive solution that is used. So part as clinical use of platelets, maintaining pH was a key parameter for retaining platelet viability in vivo when platelets were stored at room temp. The pH at 6.2 is the current standard for maintaining satisfactory platelet viability, and second generation storage containers have increased gas transport properties as well. Platelet storage and bacterial contamination. So there are major concerns associated with storage of platelets at room temp, and that is because of bacterial growth. So room temperature storage in the presence of oxygen can encourage bacterial proliferation. Sepsis due to contaminated platelets is the most common infectious complication of transfusion. So if giving a patient a unit of platelets that has bacterial contamination can lead to sepsis. An estimated 10 to 40% of patients transfused with a bacterially contaminated platelet unit will develop sepsis. In 2002, the CAP added a requirement for laboratories to have a method to screen platelets for bacterial contamination, and in 2004, the AABB also recommended something very similar. 3 FDA-approved commercial systems for screening platelets for bacterial contamination is the Platelet, the EBDS, and the SCAN system are things that we can use to check for bacterial contamination in platelets. The samples are not taken until after at least 24 hours of storage, and that is because we need the bacteria to grow at a detectable level. Potentially, it can affect availability of platelets because you have to wait 24 hours before you can give them. Pathogen reduction for platelets. So pathogen inactivation is the process of treating the blood component. The components are referred to as being pathogen reduced. So pathogen inactivation technology can help reduce the risk of transfusion-transmitted infections. There are current trends in platelet preservation and research. So there are methods that would allow platelets to be stored at 7 days instead of 5, which would help its shelf life. There's also additive solutions, also termed synthetic media, that are being tested, and procedures to reduce and inactivate the level of pathogens that may be in platelet units. There's also better platelet additive solutions, as mentioned, procedures to reduce the level and inactivate the level of pathogens that may be in platelet units. Platelet substitutes, my goodness, sorry. So maybe something that isn't actually a platelet, but something that's synthetic. New approaches for storage temperature, so like refrigerate 1 to 6 degrees Celsius, and using cryopreservation as well. Also mentioned earlier the platelet substitutes, so lyophilization and freeze drying of the trehalose loaded platelets or frozen platelets even with the cryopreservative dimethyl sulfoxide, and these can be stored up to 2 years. So that's a pretty long compared to our current 5 days.